For this lab, we're going to go over a little bit of background about Listeria monocytogenes. There are 17 different known species of Listeria. The species within the genus are non-spore-forming, gram-positive rods and are facultatively anaerobic. There are only two current pathogenic strains known, Listeria monocytogenes and Listeria ivanovi. However, L. mono is the only species that is a common threat to humans. L. ivanovi is generally associated with animal infections, mainly in ruminant species. As with all things in biology, there is almost always exceptions to this rule. A few species have been considered opportunistic pathogens. Nonetheless, this is an extremely rare case and has only been recorded a few times. In general, consider that there are only two pathogenic species of Listeria. Another interesting thing about this is that the bacteria can produce biofilms. You'll cover biofilms more thoroughly in the lecture portion later this semester. In this lab, we are specifically concerned with Listeria monocytogenes as it is the only strain of Listeria that we consider pathogenic to humans. It can grow in a wide variety of temperatures, all the way from -7 degrees Celsius to 45 degrees Celsius. Its preferred temperature range is 30 to 37 degrees Celsius, making it a psychotroph. It is also halotolerant, meaning that it can grow in higher salt concentrations than many other bacteria. It can also grow at a pH as low as 3.5 in certain cases. While Listeria monocytogenes is especially good at tolerating various environments, it can easily be killed with the recommended USDA cooking temperatures. Some things that help make Listeria stand out is that it produces catalase, lacks oxidase, and will hydrolyze esculin. Next, we're going to go over Listeria species in the environment. Listeria species can be found in a plethora of environments. However, it seems to prefer moist environments. Since this is a food microbiology course, we are going to focus the most on food. I'm sure you all have already discussed this in your lecture course, but Listeria is notorious for creating biofilms in hard-to-clean places within food manufacturing environments, specifically in drains. It can also be found in food products themselves, whether it be from cross-contamination of the food or from soil contamination. One group of food products that food safety professionals are most concerned with is refrigerated, ready-to-eat foods. These are things that can be eaten straight from the package, like deli meats or soft cheeses. You all might remember the Bluebell ice cream outbreak, which was caused by Listeria monocytogenes that was literally dripping into ice cream from an air conditioning unit. Other products like unpasteurized milk and raw fruits and vegetables can also carry Listeria monocytogenes. However, the bacteria most likely came from the soil or an animal. On February 19th of this year, El Abuelito Incorporated recalled their queso fresco products. On February 27th, the company expanded their recall to include other dairy products that were made in the same facility. Currently, there have been 11 people associated with this specific outbreak strain of Listeria monocytogenes, and at least one person has died. The Centers for Disease Control and Prevention is able to follow these cases using the PulseNet system to identify illnesses. The system allows a database that public health and food regulatory officials track outbreaks. Samples are taken from ill people and then isolated using whole genome sequencing. These sequences are uploaded to the PulseNet database to track where cases are occurring. Epidemiologists then ask ill people to fill out a questionnaire to figure out the source of the food. This specific outbreak in cheese is still ongoing and cases may rise. If you're interested in learning more about the PulseNet system, I posted an optional link in Blackboard. Next, we're going to go over some commonly used media. In food, Listeria is usually found at low levels, so a few enrichments need to be done. The University of Vermont broth is the primary enrichment broth. It's a selective medium which contains nalidixic acid and acroflavin. The nalidixic acid inhibits gram-negative bacteria, while the acroflavin suppresses other gram-positive bacteria. The esculin is hydrolyzed, which is when you see that dark color as displayed in the left test tube. Fraser Broth is a selective medium which is used for the secondary enrichment of Listeria species. It contains nalidixic acid, acroflavin, and lithium chloride. Lithium chloride is a kind of salt and is used to inhibit the growth of enterococci. Listeria species are known to be salt tolerant. Listeria species hydrolyze the esculin to esculatin, which reacts with the iron in the ferric ammonium citrate, which results in a black precipitate and a visible positive reaction. In Mock's agar, or modified Oxford agar, Listeria species are able to grow in the presence of antibiotics, which are the top two listed ingredients here. The esculin and ferric ammonium citrate differentiate the colonies with a black precipitate. The protocol we will use in lab is to create a 1:10 dilution of the food sample in UVM broth. The sample will be stomached and incubated for 24 hours at 30 degrees Celsius. We will then take 1 ml from the incubated UVM enrichment and add it to a 10 ml tube of Fraser broth. This will be incubated as well. We will then take a loop full of the incubated Fraser broth culture and streak it onto a Mock's plate, which will then be incubated again. Isolated colonies from the plate will go on for further biochemical testing like API testing. One could also conduct whole genome sequencing to be even more specific about the strain. One method of biochemical testing for Listeria monocytogenes is the Analytical Profile Index, or API. So what is an API test? It's the classification of bacteria based on rapid biochemical tests. Various types of API strips for different types of bacteria are available. Some examples are Listeria, gram-negative bacilli, lactobacillus, etc. The Listeria API test differentiates Listeria species. The presence or absence of acrylamidase, which is a DIM test, which stands for differentiation inocua monocytogenes, hydrolysis of esculin, the presence or absence of alpha mannose, or acid production from the following, can all differentiate different species of Listeria. As you can see here, the DIM test is the only one that really differentiates L. monocytogenes from L. inocua on the Listeria API test strip. Here's just an example of the Listeria API test with the Listeria monocytogenes on the top and Listeria inocua on the bottom. On Wednesday, you will have a video come up of how to perform an API test strip. It won't be for Listeria because I couldn't find a very good video, but it will be to show you how the different testing methods work on a different kind of test strip.